Well, welcome everyone to, I don't know if this is the penultimate or the last seminar of the season, or maybe there are a few more to go. But anyway, I'm very pleased to introduce Dr. Rosemary Walzem, who's a colleague and friend of mine, and just a little bit of background how we became acquainted was we both sit on the Intervention Testing Program Access panel, which is the one that reviews all of the proposals for testing drugs and other agents, nutraceuticals, and so forth that might influence aging in a positive way. And I was impressed from the get-go. We've been on this since 2010, I think, with Rosemary's breadth of knowledge about phytochemicals and other agents, many drugs as well, and kind of set up a respect for her that ultimately led her to invite me to College Station to speak to the undergraduates about the biology of aging. Yes. And that was really an interesting experience to go from a place that has about, I don't know, a couple hundred graduate students to a place that has
52,000 undergraduates.
Yeah. So it's very, very interesting. So
absolutely, every single one. It was just terrible in the room.
They were all over the place. But so I wanted to reciprocate and bring Rosemary here for a couple of reasons. Number one, her area of interest is a little bit out of the box with respect to aging, but I've always been someone who likes to be out of the box and bring people together from diverse perspectives. Particularly in aging research, I think we benefit greatly from learning about all kinds of other things that potentially can impact on aging. And her background is in nutrition and comparative biology of nutrition, actually, and many of us are interested in the comparative biology of aging. So that was another reason. Another reason was a bit of a selfish one. I wanted her to become acquainted with our aging program here, and since she has access to 50,000 students, or maybe not quite that many, she could serve as a potential conduit for new graduate students coming here. But anyway, just a little bit more background about Rosemary. She did all of her training at UC Davis in nutrition. Yeah. And so she got a bachelor's degree there, a master's degree, and ultimately a PhD. And she did her early professional work there for about 10 years before being recruited to Texas A&M in College Station to head up a new nutrition center there. And subsequently, she's carried on. She's a full professor there and has done a lot of work in chickens and other avian species, as well as human crossovers.
And mice.
Yeah.
Don't forget mice.
So anyway, without further ado, I'd like to welcome you to our group, and we're very informal, so this could go on a little longer than the normal seminar, or a little shorter.
Okay.
Thanks.
Let me turn myself on here. There. All right. Well, thank you for having me, and this is really very pleasant, and I always enjoy talking with Jim and aging intervention, and that dynamic has certainly expanded my perspective. So this probably is a little bit different, but I picked these. Jim gave me some guidance on the sorts of things, and so we're going to talk a little bit about caloric restriction at some point, and you will see how that involves with chickens. If you have any questions about chickens, you can ask me chicken questions. I know many of you have questions about chickens. But so it turns out that in agriculture, and I do kind of go between like clinical, I'm a registered dietitian, so I do human trials, but then we do a lot of animal models. So in agriculture, it's real important. What costs you money when you're growing a piece of animal is the food. So the animal is the least of your cost. What costs is the food. So they're always very interested in efficiency and getting the most out of the feed that they feed. And there's no place where genetic selection hasn't had a bigger impact than in poultry. So poultry, if you really get into poultry and how people manage poultry, there are particular strains of poultry for different things. And let me start this off by saying birds basically already start life in a very constrained energetic context. And so I think they are holistically just really concerned with energy. Good, this works. So, as we know, birds are oviparous, which means that the embryos incubate separately from the hen in a hard-shelled egg. So if you think about that, I mean, with a placenta, you can have mom can walk around, mom can eat, mom can keep feeding. No, the bird has to have absolutely everything they need in that egg from the get-go for a successful incubation. And if it isn't there, you don't get a bird. So yolk formation, yolk is really where most of the energy comes from for the developing bird embryo. And it is a multi-tissue, highly coordinated process that has a lot of places it can go wrong. And so that's sort of what I've studied is partially is where it goes wrong. So the embryonic requirements for hatching, and as we go forward and I talk about different kinds of chickens, realize pretty much chicken eggs come in, I mean, I know there's large, there's extra large, there's peewee, we can talk about why that happens, but for the most part, they're basically very similar whether a bird ends up being a meat bird or an egg bird. So they all start out the same. They need, of course, some glucose, they need protein, they need their essential fatty acids, they need vitamins and minerals, but mostly they need energy. And when they actually measure this, and there are guys that spend their whole careers measuring this, believe me, when you incubate eggs, the embryo and the heat that it produces from the inefficiency of its metabolism ends up being about 60 calories. That's what it takes to get a bird out of an egg. So when we look at an egg and we see what's in it, we see that the egg white has about 18 calories. That's all going to be protein. So for the most part, that's actually going to tissue, not energy. The yolk is what has the calories. It has about 70 calories. Okay. Now, the yolk itself is actually a sack of lipoproteins. So what egg yolk is that it is a whole bag full of triglyceride-rich lipoproteins that are made in the liver and then transported down to the ovary and selectively taken up and deposited into the egg yolk. So macromolecules of triglyceride-rich lipoprotein, and this particular cartoon, I know this is not the usual, ball with a 4-dimple rubber bulb on the side for ApoB, which I hate. This is the correct configuration. This is ApoB. It's a very large protein, and it adopts this folded conformation on the surface, so it's embedded in the phospholipid, and it is the crisscross right here that is the receptor recognition that pulls it into the yolk. So ApoB is non-exchangeable. It is a structural protein, and it is the receptor ligand for this particular particle. And it is the actual structure that allows for the transport and regulated distribution of lipophilic nutrients within the polar environment of the blood. Okay, so some of you I know look at diabetes and things like that involve some lipoprotein metabolism, so this may be very familiar to you. In most mammals, the way this works, I guess I'll say all mammals, the liver, let me go back. The liver will make a triglyceride-rich lipoprotein and secrete it, so you'll get a very low-density lipoprotein, a VLDL, and it'll come out in the periphery, and lipoprotein lipase, which is present on peripheral tissues, will chew back the triglycerides. Triglycerides will be taken up, and you'll get a progressively smaller and more dense particle form from this original triglyceride-rich one. Initially, you'll get an intermediate density lipoprotein, and then once that particle actually gets a chance to go second pass at the liver, you'll end up ultimately with what we call an LDL. So this is sort of the normal progression that people think about with lipoproteins. Here we're looking at particles that are anywhere from 70 to 80 nanometers. LDL, generally speaking, they're really small. They're 21 nanometers, 25, 24, we're into pattern B and better things. Okay, so the bird ends up having an issue. And we'll get to pictures of actual ovaries, but when you look at a bird's ovaries, they have yolk follicles. So there is a follicle inside which a yolk develops. So it is surrounded by tissue layers. These tissue layers, there's the theca, which then has small blood vessels that run through it and carry, here we have red and white cells, and then these little tiny yellow dots are the VLDL. And we'll talk a little bit more about them in a minute. We have granulosa cells. So these are hormone-producing cells that help maintain the follicle and actually drive the whole reproductive system. They produce estrogen. And then there's a granulosa basal lamina, which these particles must pass through. And on the surface then of the oocyte, there are actual yolk receptors that are basically ApoB receptors. And so this goes through the whole endocytic process very like any sort of lipoprotein uptake in the liver or macrophage. Now, the thing that's really fun about egg yolk, these yolks grow. And so this is a continuous stream of particles coming out of the liver that is being continuously taken up by these receptors, which means that the yolk grows in layers. And so if you're looking for an undergraduate science project to make them go, ooh, you can hard boil an egg and cut it in half and then stain it with silver stain, and you can end up seeing these layers. Alternatively, you can feed your chicken on alternate days red and blue dye, and then when you hard boil it, you'll end up with red and blue rings. That's also fun to do at Easter. Okay, so the thing that I would like you to see is that these particles that are coming into the yolk have to pass through this cell layer in order to actually access the receptor that's going to take them into yolk. So here we see chicken follicles in development, and they start out when the bird hatches. It has all the ovarian follicles it will ever have in its entire life. They're small, tiny pinpoints. Once they actually become reproductively active, then those follicles begin to grow, and you'll get what they call small whites, and there'll be many hundreds of those. You'll get small yellows, and there'll be more of them. There'll be hundreds, but not several hundreds. And then these are then selected as they begin to actually really deposit yolk in them, and they enter what they call the hierarchy. So once they enter the ovarian hierarchy, they are committed to ovulation. Now, prior to this, every day a certain number of these follicles will be recruited and started on their developmental path. They die off until you get to that one that gets recruited. And it comes in, and then over a period of 8 to 10 days, it will achieve a full egg yolk size through this deposition of all these VLDL coming in.
So before puberty is this happening?
You only have, and you can't even see them, these very, very tiny. They're there, but they're microscopic. So absolutely every ovum that bird will ever have in their life is there at hatch, but it is so tiny until reproductive stage starts. It's hard to see. It's like a little nub in there. So that's one thing. You have this, so it's a very different thing from mammals in that you have sort of this time sequence that goes on. You have recruitment of a cohort of follicles with development, and at each developmental stage, some of them fall off. And so one of the big questions that people ask, not me, but other people, what is it that decides whether you're in or out? Why do you progress and others in your cohort get wiped out? It's a question. Now, when you look at these yolk follicles, you'll notice that they are covered by this fine network of blood vessels, right? So this is that theca granulosa layer. And then there's this clear spot. This is called the stigma. This is an avascular zone, and this is what actually ruptures and releases the ovum into the oviduct. So that way the bird doesn't bleed to death when it ovulates, right? Rupturing the stigma is a proteolytic event. It's also a controlled oxidative event. So once this ovum, and you'll see how they're different sizes? So if you're cutting this out and you're somebody that's trying to say something about where this bird is in life reproductively, you can rank these. This would be the F1. This is the follicle that's going to go out next. F2, F3, kind of a call here. If we go to the bigger picture, there's F4 and F5. So this will come out tomorrow, the next day, the next day, the next day, the next day, so that you can see almost a whole week's worth of development going on. So once ovulated, the ovum then passes through the oviduct, and it's at it spends very specific amounts of time in each region, acquiring first a thin white and then a thick white. And this all takes, moving from here to about here, takes maybe 2 hours. And then we hit the shell gland. And in the shell gland, you now have a yolk with thin white, thick white. And when you break open an egg, you can kind of see that different thick and thin white. And then there will be a membrane around that whole bag of stuff. At this point now, this egg sits in the shell gland for 20 to 22 hours and gets its shell put on. And basically, the way the shell gets put on, it's like powder coating metal. So that entire structure with the yolk in the middle and all these layers rotates. So when you break out your egg and you see those little squiggly chilazas, that's because that thing's been turning and turning and turning while it's been acquiring its outer coat of eggshell. That part, I don't know. I believe it's just muscular. So I don't know if there are actual cilia involved. This entire organ actually, while it's very distensible, and when you take it out, you have to be careful or you could tear it. It's that delicate. But this is actually quite muscular, and I believe it's primarily just a muscular, it muscles it around.
What stage of this nuclear growth is
fertilization? Fertilization, so up here, what is not cut out for your benefit is there are what they call sperm storage glands up here. So fertilization happens immediately after ovulation, because once the thin egg white goes on, sperm ain't got a chance. So there are sperm storage glands that surround the infundibulum. So as soon as the ovum comes there, if the bird is fertilized, then those sperm will come out. It's a coordinated hormonal event. And eggs are actually fertilized by polyspermy. So sometimes it's a little hard to tell who got in there. But so that's where that happens. So actually, if indeed the bird it's a fertile flock and you're having fertile eggs laid, by the time the egg is laid, so once it's acquired its shell, moving from here to here is about 15 seconds. I mean, it's just out. That's oviposition. At that point, the embryo has 70,000 cells in it already. This is one of the reasons that transgenesis is so difficult, is that, capturing a yolk and fertilizing it and doing it, it gets complex. So there's a whole different process than what we do with mice. Absolutely. Table eggs. An industry is born. No, it's one of the greatest. They historically, when get to the philosophical poultry scientists. So I don't really consider myself a poultry scientist, but there are guys that are deeply poultry scientists, and they have whole philosophies about these things. Evidently, there is a Sanskrit poem from 4,000 years ago, an ode to the hen. Oh, I love her so, she lays an egg a day. And this is very unusual. Most birds lay clutches of eggs. So, they'll lay 10 and that's it. Some of them are indeterminate layers, and they will lay their 3 or 4 eggs, and if you take them away, they'll lay some more. But for the modern breeds of poultry that lay eggs like these guys are going to be laying it, it's actually considered sort of a mutation. So
we think they were selected for that.
no, we think it was just a lucky happenstance that some bright human went, damn, I'm keeping that one. And then, then it went out from there. Okay, so let's get back to this energy question. So I told you about VLDL and how in humans it gets smaller and smaller as we deplete the triglycerides in the peripheral tissues. So how would that work for a bird if it worked that way? And initially in the 70s, there was a lot of lipoprotein lipase shown to be living or existing on the ovary cells. So they thought that that's how it was, and then somehow the triglyceride was reassembled inside the yolk. It's before Brown and Goldstein found the receptors. Okay, so an average VLDL has a diameter of 70 nanometers. It's about 65% triglyceride, which means that it's got, and I did some math for you here because there's something on the order of 7 times 10 to the 18th VLDL particles in an egg yolk. Okay, so lots of them. But this can contribute about 36 calories. So that's kind of that's okay. We're right there. The only problem is that granulosa basal lamina excludes everything that's bigger than 30 nanometers. You can't get, so there's a mechanical sieve there, okay? So let's look at a small VLDL that's, so maybe it's more like an IDL. It's only partially been ripped apart. It's down to 55% triglyceride. That now would only give 31 calories. So we're running a little bit of an energy deficit already. So this is a problem. And if we look at where most triglyceride-rich particles go in a normal mammalian system, you can see very quickly that you would not be able to actually hatch a bird if you had the same kind of lipoprotein system that mammals have. So it's a big problem. So luckily, the birds do something different. And what they do is that in a normal laying hen, once estrogen hits, and this is actually a very beautiful genetic program, you can give a bird a shot of estrogen, a chicken. Inside half an hour, this whole liver is rearranged and produces a completely different particle. It's still very triglyceride rich, but instead of 70 nanometers or 60 nanometers, it is now 30 nanometers and small enough to go through. Sorry, there's somebody. It's that same siding salesman that called me earlier. Okay, so we now have these 30 nanometer particles. So they're small enough to get through, but you know, what are they? It turns out they are rather different. So instead of regular sort of 60, 65% triglyceride, it turns out they're actually quite triglyceride rich. They're about 67% triglyceride. So when people tell you egg yolks are super rich in cholesterol, it turns out as far as lipoproteins go, they're sort of uniquely cholesterol poor. They're actually quite triglyceride rich. And they're small in size, so of course they're going to have more phospholipid because of the surface to core ratio. They do have a little bit of cholesterol, and this has probably got some genetics in it that I'm not going to get into. But for the most part, they're small. And so this is one thing we did spend some time on early in my career. We did a laser particle sizing, and we were looking at, this is dynamic light scattering. Roosters, they have big particles. You can see they're around about 70 or 80. Some of them go up to 120 nanometers. And then hens, the other thing I'd like you to notice is not only are they smaller, but see that they're quite uniform. So they go from being this tailed polydispersed profile to quite a monomodal system. And there's another change, and let's see if I have a picture of that. Nope, let's go back. So let me just tell you then, the other really crucial thing about laying hen VLDL is that they have a protein in them called ApoVLDL2 that specifically, what it ends up doing is it limits the amount of water in the surface of the particle, so lipoprotein lipase cannot hydrolyze it, because if water can't be there for the reaction, then it doesn't happen. And so we actually used NMR to show that it's excluding water from the surface of those particles. So these particles come out, they're triglyceride-rich, they're small enough to go through that mechanical sieve at the granulosa, and they are resistant to any sort of lipase action. And so they are very effective at delivering triglyceride energy to the embryo. So it's a completely different evolved system to make sure those little chickies hatch out. Good for us. Okay, why is this important, and why am I going to talk to you more about energy? Let's just point out how big a deal chickens end up being in our usual dietary life, and one of the reasons why. So they've been selected heavily to be highly efficient, and while they are good sources of protein, a variety of unsaturated fats, vitamins and minerals, and all that sort of thing, one of the reasons that we eat, what is that, 2.18 times 10 to the 12 pounds of chicken per year globally, is that a gram of protein from chicken meat costs less than a penny. Whereas a gram of protein from salmon costs about 8 cents, and a gram of protein from a ribeye, and I only went with select, I didn't even go prime, is about 9.5 cents. So it's cheap. This is why they can add chicken to your salad for $1. Eggs, same sort of thing. And in fact, the protein from eggs is even cheaper, depending on, this is your regular Kroger dozen, and this is pasture raised, okay? So even those really expensive, oh my God, $6 a dozen type eggs, they're still fairly reasonable. Cheaper than salmon anyway. So high quality, inexpensive. We in the poultry industry also like to point out, we've been determined to be the least environmentally unfriendly source of animal protein. So we're pretty much golden. What? Yeah?
Chick hatches, it has to immediately be hardwired to start feeding.
You had anticipated me. Yes. Here's what genetic selection for meat has done to chickens. 1957, at 56 days of age, the average chicken weighed 905 grams. By 2005, that same chicken now weighed 4,200 grams by 56 days of age. The geneticists for poultry companies tell me there's no down on that. They can keep selecting. They're going to just get bigger. And so the joke at the poultry meetings is that, well, someday in about 20 years and 15 minutes, we're going to break open an egg and it's going to just magically go to 5 pounds. Okay, so they've done this. And you can see, here's a chick and the ones that are selected for eggs. They start at the same weight. They started at about 44 grams. And so by 28 days, they're almost 1.5 kilos. Well, they just, yes, they are big birds when they're done. But, and so if you go to the grocery store, and I don't know if you can even buy them anymore, but those chickens in a bag, the whole chicken, that bird's only about four weeks old. So it's a baby. It's a baby animal. So there are 3 poultry genetic companies in the world, and they are very private about their birds, okay? So the other thing you run into is that for meat birds, basically the most they live is 10 weeks, because at that time they're all processed. So the breeder birds that we'll talk about in a minute, if they're not laying eggs, they're gone. So, no, nobody looks at that because that's not necessarily what they care about. But if they had a little more foresight, I think they'd be very interested in aging and how long could we make them go and what do we need to do instead of just selecting for growth, if we selected them for productivity. And so actually the first step in that direction that I've seen as a positive is that they no longer talk about yield in pounds of meat per bird or percent yield that way. They are now talking about meat produced per hen. So they are now encompassing, bringing in how many eggs does that hen lay before she gives out, and you'll see why that's important.
So how long do chickens live?
This is sort of a guess, but if they're just sort of regular standing around chickens, probably 13 to 15 years. So table egg birds, you can see that they too have been selected so that they have also increased their productivity. And in this next little graph, I believe, so here we see pictures of sort of the standard chickens that are used commercially. We have the very beautiful W36 Highline laying hen. And over here we have the Cobb 500, the most efficient meat bird in the industry. Okay, so those are all taglines, you now know. So what you can see, here they look about the same size, but they're not. In a layer, and that's what they call them, they're layers and they're breeders. So in a layer, the production cycle in weeks, how long are they going to lay eggs? A layer bird will lay for 90 weeks before the eggs get to a point where they aren't going to use them. A breeder has about 44 weeks, so half the time. In her 90 weeks, the layer is going to lay about 500 eggs. And the breeder is, if you're a really good manager, going to lay 175. Body weight on these layers is about 1,500 grams. That's their absolute adult body weight. And the breeder hens are about 4,000 grams. And we will get to why it is that that's all they weigh. Feed intake per day, and in the layer hen, this is spontaneous feed intake, is about 95 grams. In the breeder, to achieve these statistics, she is fed about 166 grams a day. Okay. So this is a little bit disingenuous of me.
Quick question.
Okay.
Just in terms of the commercial aspects, the breeder is used to lay eggs until she's used for meat. Is that?
Yes, but the meat that she would get used for is probably something more along the lines of your cat or dog, or perhaps canned meat. So.
Is she also then used if she's killed earlier for the meat that we eat? Or who gives us that meat?
That's going to be those 4 to 10-week-old birds per baby.
And there are neither of these strains?
Well, it's this strain. It's her chicks. So breeder flocks produce fertile eggs that are incubated and hatched for production. And they're grown out to a certain age for meat, and then they're slaughtered. So these birds, if they don't lay eggs, you don't get meat.
You don't use those eggs for eating?
No, you don't use these eggs for eating. These eggs, okay, so here you go. Now, I know mice people are going to have as skewed an opinion about animal pricing as I do. So when I do chicken work, when I used to do mouse work, a mouse, $20, no big deal. Not one of the really expensive ones, not one of those ones that cost you $200. When I ask for a breeder hen, which is a really expensive hen, my farm guy says, "Oh, Rose, they're probably going to charge you like $7 for that thing." I'm like, "Dale, that's okay. I can pay $7." So, and then here, if you're a scientist on a budget, turns out that estrogen trick works as well for males as it does for females. And on these table egg birds, of course, they're only interested in females, right? So if you live near a hatchery, you can get all the male birds you want for free, because they're just going to kill them anyway.
You're thinking words like entropy, and the breeder birds just looks to me like this is not an arithmetic progression, it's a geometric progression, isn't it?
If they could get them to grow logarithmically faster, they would love that. That's sort of the genetics.
It's somewhere in between the two.
Yeah, and I'll show you better growth curves. I'm going to show you some growth curves that are out of the breeder's standards. So these birds come with instruction manuals if you're going to really grow them. Okay, like 15, 20-page things: how to build a house, how to feed them, how to weigh them, everything you're supposed to do with them. Okay, so the table egg birds, I got into this because table egg birds used to have a real problem. They got this thing called fatty liver hemorrhagic syndrome, and you can see some of the lesions here. And so these are chicken livers, and you can see they're very pale because they're laying eggs, so their livers are actually very full of fat anyway. And periodically they will get a rupture, and so this is a bleb of blood underneath. This is a bruise. This one has got some old, and with old hens, you will pull them out and you can see scarring or old bruises that are reabsorbing. And when this really happens is you have birds that are really laying eggs. It's a very energy-intensive process. They're eating, and suddenly the temperature goes up. They used to hold conferences on this at A&M, okay? This was like a huge issue in Texas back in the 50s. I mean, when I was going through the old literature, all those guys at Texas A&M, lots of papers. So as soon as the temperature goes up, birds get warmer, don't need as much energy, but they don't stop eating fast enough. And their livers would suddenly go with fat. And then if they were laying an egg, with the pressure of oviposition, they end up blowing out a vessel. And so we tried to do this because spontaneous cases, I mean, you got to stand around, wait for it to get hot. Farms don't really like you to come in when they're having a health issue. And so we did purified diet fed, because I was nutrition trained, and you got to have a purified diet, right? So these are these lovely W36 leghorns, and I gavage fed them 3 times a day so that we got them up to 150% of their usual food intake. And at that level, they in fact manifested all the symptoms you would see with fatty liver hemorrhagic syndrome. And this is just a reticulin stain of their livers that came out. And you can see here's normal liver with its nice hepatic cords, nicely organized and all that. And then here's the one that is overfed and very fatty and completely disrupted. And you can see that the reticulin that actually holds these livers together is getting blown apart. And so that was really the issue. So at that point, I was a postdoc, and I was like, man, feeding these chickens 3 times a day is really a deal. That's this is a drag. So one of the things, now I will show you this. So how big of a deal was this as far as what happened in that amount of time? You can see I could only do 18 or 17. They gained 325 grams. They upped their food intake by 50%. Their liver weight doubled. They gained some abdominal fat. Their ovary got to be a little bit less. Their egg production went down. And partially this is because, 3 weeks, I'm done. This is too long to be out there 3 times a day doing these birds. So we got an effect. I stopped it. A lot of shell defects. And the diameter got bigger. So we thought, well, maybe that's it. That's why we're getting poor egg production, is these particles just get big, can't get in, and so it all falls apart. So we were pretty happy with that answer. These are weird ovaries from various things. So here's this normal one, and you can see you have the difference in size. And you have one that's called expanded, and the British used to report these all the time. British people. So they would report these, and I never saw these. It's like, what are they talking about? What do you mean it's messed up because of an expanded hierarchy? We see collapse. So we would see this sort of thing where you see they're all dark. You can see that they're sort of falling apart. You can see this one's completely regressed. This here is called a post-ovulatory follicle. So it's just sort of left over here. these all got retained. They have various, I don't know what that is. It almost looks like bile or some kind of, anyway, they're really messed up. It's pretty clear this is not going to produce an egg yolk any of us would want to eat. So mostly what we saw was atresia. That's what we saw. So I never understood this expanded hierarchy thing, although we'll talk about that in a bit. So how different are broilers and layers? These are breeder expectations for growth. Juvenile growth rates differ markedly in meat and egg type birds. So here's this broiler bird, and it goes from being about 40 grams to being close to 5,000 grams inside of about 10 weeks. So this is market weight. This is where their life ends. Now, layer hens, of course, go on longer because you want them to lay eggs, so they have to be grown-ups. But you can see that these birds never get past 1,500 grams. That is their body size, and in fact, some strains only ever make it to about 900. They tend to be smaller eggs. So there's this huge difference. And their spontaneous feed intake differs markedly as well. So again, the layer hens are eating much less food per day, and they top out at about 95 grams a day. If you let the broilers eat, as they are wont to do, let them just eat how much they're going to eat. They eat anywhere from, well, I've topped this one out at 250 grams, 265, 270, not unusual. So they eat a lot more. And they do, pretty much. If you let them, they will stand around the trough all day and just eat. So the whole notion of giving them an outside lawn to run in, they look at that thing, it's like, that is not in the direction of the feed trough. That is the feed trough. I'm walking that way. So, just saying, they're hungry birds. Okay, so it turns out if you, in fact, let those broilers eat like that and grow to adulthood, basically by about 30 weeks of age, they're dead. They die of obesity, hyperglycemia, heart failure with arrhythmia, blood pressure issues. They do not do well. So, and they certainly don't lay eggs, not in any sort of context that would be commercially profitable. So they've empirically determined that what they have to do is they have to limit the growth of those birds in order to get them to lay eggs. So this is where we come to energy restriction again. So here we see what their growth would be if they were allowed to eat and you were raising them for meat. This red line now is what broilers are grown to. This is the spec that you have to hit if you're managing them for egg production in order to get them to lay until they're at least 60 weeks of age and you can get enough eggs out of them to make money. Now you will notice this is a remarkably lower growth rate. Okay? Now here's this growth rate, the same for the layers. They lay to 110 weeks of age or so. Now the feed restriction, and they like to do this with the broilers because they lay longer if they start them later. So they will restrict them to the point where they don't start laying until they're about 25 weeks of age. The regular table egg birds, they'll start up 19, 20 weeks of age and just go and go. So I had a master's student, and we were trying to look at these lipoprotein particles before they lay, at 50% lay, at peak of lay, and then when they quit laying. 2 years into it, she said, I got to go. They're not stopping. And it's a master's. I have to leave. And so they came down to 65%. So I said, fine, write it up. We'll be done. Okay, so to get them to delay their growth or reduce their growth this way, this is how much they restrict their feed. So you can see that they are now, instead of letting them eat their 250 grams, so they restrict them heavily. And the worst restriction comes in here, where these birds are really trying to grow genetically. I mean, they're really trying to grow, but they're being really starved and held back. So if we're dealing with a human population, WHO would say they're stunted and wasted, right? So But it works for these birds in terms of egg laying. And you can see this is some nice data that came out of Canada way back in '92 from Frank Robinson's lab. So full-fed birds laid 122 eggs. Only 103 of them were settable, which means that they were fertile and not double yolked and not damaged and all that. You can see that their yolk weight was higher. Remember, yolk is what amounts to sort of an adipose depot, right? It's fat. That's what's in there. They're delayed. So because the ring number went up, that means they were actually in the ovary longer. So their actual development was delayed. Their ovulation was delayed. 8 out of the 20 birds studied had regressed ovaries. So that fit with mine, not what those English guys were saying with that expanded hierarchy. And they had gotten a little bit fatter. So obesity in a breeder hen, 8% body fat, we're talking, well, it's abdominal. If you have a fat pad that's 8% of body weight in the abdomen, this is a fat bird. So this just tells you how lean the other guys are. So when they restricted them, you can see they got a lot more eggs. They were all settable, a little bit smaller yolk, but they were moving through the whole ovary quicker. No regressed ovaries, really, just 1 out of the 20, and they had less fat. So it really does make a dramatic difference in terms of their reproductive longevity.
Rosetta-type ovaries, if you restrict their diet, is that what makes them produce these multiple they're all hierarchy?
No, the restriction will get them into a more ordered hierarchy, so you will have an F1, an F2, and F3 that are very clearly different in size, and one will come out, and then the next day the next one will come out because it'll be a little bit bigger.
Better.
Yes, the feed restriction does. So, but it is true, these birds are pretty hungry, and so this is, when they place the food for the breeder hens, there's a fight at the troughs. It's a big welfare issue in the industry that, in fact, how do you get this feed out to everybody all at one time? There are pecking orders in birds, so you have dominant birds that are going to muscle weaker birds out and beat them up and stuff like that. So, and I told the students that really what they do to try and manage this, the notion is, well, if we skip a day, then we give them twice the amount of the restricted food every other day. Then the days that they do get some food, they have more time to eat because there's more food. I think skipping a day is kind of tough. At least I don't like skipping a day.
But the dominant birds are going to end up with more food and they're going to make less eggs.
Yes.
And the birds that have been starved and that are on the bottom of the hierarchy are going to make more eggs.
If they get enough energy to actually come into lay. Okay, so I will skip over that because I'm being very long-winded about my chickens here. So we did a study where we greatly simplified things. Instead of intubating layer hens, we just went ahead and got broiler breeder hens, and this is an acute model for extreme overeating. So we take these breeder hens, we bring them into our facility, we let them get used to our cages for a week or so, we monitor their egg production and see who's laying at what rate, and then we let some of them just eat. So we unrestrict them. We just give them a full feed trough. They're all individually housed, so there's no fighting going on. And then we see what happens to them. And so when we do this was back in 2006, and this is where we really demonstrated that in this acute setting, you basically end up with a bird that is hyperglycemic, hyperleptinemic, it is hyperinsulinemic. It's really almost a type 2 diabetes model. So here is just an indicator of the feed. So in these birds, the breeder recommendation was a little bit less than 160 grams a day, and so the control group was maintained at that. And then we had two groups. One was just ad lib, this is F, and then twice normal allotment. And you can see that basically after a little bit of excitement with the real ad lib, they came back down. They get a little bit sick looking. It's like a Thanksgiving Day effect a little bit. But they come down to where they basically will eat this amount every day. And there was really no difference between the two, so we pooled them. So the next set of statistics, you'll see 15 versus 30. And what we saw was a difference in, so inside of 10 days, this is a 10-day thing, they gained 500 grams of body weight. They doubled their food intake. Their liver became close to 4% of body weight instead of about 2%. Fat went up a little bit. Ovary weight went down. Eggs per day went down. Follicle yolk weights went up, and atretic follicles went up. We then looked at it because we saw that about half the birds really went out of lay and about half of them didn't. And it may have to do with where they were on the energy status curve in the flock. But what we saw was that the ones that were able to maintain a normal ovary stayed much closer in terms of all of these. They still gained 500 grams, but their ovaries stayed normal. They didn't have quite so much fat, and so they just stayed closer to normal, and the ones that had the abnormal ovaries were clearly fatter. So what's going on with that? This just shows you the difference in glucose. You can see that it went up from 2 mg per mil to 3, and that insulin doubled. So glucose went up even though insulin went up, and so that's at least my definition of diabetes. Leptin, and there is a fight about avian leptin. I know this, but we sent them the samples blind, and they sorted perfectly. So whatever the heck they're measuring, it seems to be going with treatment, so we reported it that way. But you can see abnormal ovary even higher. Insulin even higher. Glucose even higher. And then the lipids were changing in terms of what was going on, and in particular, they didn't seem to be able to desaturate fatty acids that were being made de novo. And we saw apoptosis in the granulosa cells. So this is just some incubated granulosa cells with either perpidium iodide, which is shows up as red and necrotic, or annexin phos, which is apoptotic. So 0, normal ovary. I don't know if you can really tell, but these were brighter. We had both more necrosis and we had more apoptosis. So what was going on? At the end of this, our real questions were, we'd shown they developed insulin and leptin resistance, fatty liver and fat mass gain. They were obese. They had larger yolks with more rings, so that was delayed. So that was our question. Why is ovulation delayed? What could do that? We thought there was probably altered cell signaling as a potential mechanism, perhaps lipids. And so really, at this point, this is my student Chen. Went back to Taiwan and took up his own lab, and he took this project pretty much with him, although I keep working with him. So he did a postdoc down at Baylor, and now he's over in Taiwan and a big muckety-muck over there. So with his lab, we started asking these questions, and just remember what it is that yolk follicles got. They have the granulosa cells that are for hormones. There's this stigma, this avascular region. Leukocytes have to participate. They have to bite through that to get the actual ovum to ovulate. And then we have liver. So let me go through this kind of quickly. Bottom line, what it came out to is that the liver was cranking out lipids so fast that one of the things that we got was a lot more palmitic acid because they weren't desaturating it. And through the palmitate, we got a lot more of ceramide, both through de novo synthesis and also through sphingomyelinase. And we did some experiments to inhibit this step, and we did some experiments to provoke that step, and we showed that what's happening when we actually isolate the VLDL. So this would be your bioactive lipids guy. What we showed is that the liver was making VLDL particles that were ceramide and sphingomyelin rich. And so that's going down to the ovary. And remember, those particles resist hydrolysis, but they aren't impervious. And so we believe that what was happening is that the VLDL from the liver was delivering more ceramide to the granulosa cells, and that was part of the reason why we were seeing the apoptosis. So this is just the experiment where we did that. We had these birds, Arbor Acres breed, 60 of them, 23 weeks old. They were laying pretty well. Adapted them and then just let them eat for a good long time. And really, other than what I was telling you about the ceramide and sphingomyelin, so tag, sphingomyelin went up in ad lib versus restricted. At that point, at 35 weeks, so this is now about 10 weeks after they started this, estrogen was staying the same. A little bit longer when they're really falling apart. So at this point, in restricted birds, no bird had died. Even though it's hot in Taiwan. 9 of the 40 in the ad lib group had already died. 50 weeks, so a little bit further on yet, egg production is down quite a bit. Remember, these guys started out at 87. So the ones that are restricted and doing what they're supposed to, they're already senescing. They're already down to 62. And then in the restricted or the unrestricted, they're down to 24%. They're really looking bad in terms of their lipids and glucose. Estrogen is now falling down, which means they're all going to have ovarian collapse because they aren't getting the estrogen they need to maintain their reproductive tract. And 17 of the 24 are dead. So, and he went on, we did some more studies, and he's the one, his lab showed that the arrhythmias in the heart and all of that. So, tissue ceramide increased in liver, VLDL, follicle wall, granulosa cells in the ad lib hens. Ceramide increased both by synthesis and by remodeling. This was true in both the liver and the granulosa cells, and we saw a whole lot of tissue inflammation, increased IL-1 beta in the liver, the adipose, ovary and muscle, and reduced cell survival signals, PARK, RKRP expression in granulosa cells. So this is the experiment where we tried to look at it in culture. So cultured granulosa cells, if you give them a high physiologic palmitic acid, you increase ceramide formation in the granulosa cells. If you add this inhibitor of de novo synthesis, you can improve their cell survival, and it would also inhibit the inflammation and apoptosis. So we're pretty happy that we understand that. The other thing that happened, you can see, okay, these are yolk follicles, so it's really hard to cut through yolk, even if it's frozen. So that's why that's ugly down there. The really important thing to look at is in this surrounding follicle layer. This is in ad libitum, and all these little arrows and blue dots, this is a whole bunch of leukocytes that are moving in. So birds have heterophils instead of neutrophils, right? So heterophils and monocytes moved in. In the restricted, you did not see nearly as much leukocyte infiltration. This is kind of a crappy antibody, but it's the only one we have for chicken monocytes, macrophage lineage. And what you see is a lot more monocyte macrophages coming in and trying to be there, but unfortunately they got lost and they were pretty ineffective. So PMA responsive burst in these leukocytes coming out of ad lib fed birds. It was reduced in heterophils but not monocytes. And then the IL-1 beta secretion in response to LPS was reduced in both, but more importantly, the I can never say this word, the matrix metalloproteinase activity in response to LPS was also reduced. And so what we think is there's more yolk rings there. Follicles aren't getting out because basically the leukocytes can't find their target, and when they get there, they can't actually chew very effectively, so that you can't rupture the stigma and the follicle can't come out. And I would love to do some sort of on the surface of the follicle thing to show all these confused monocytes and heterophils and how lost they are, but we haven't gotten there yet. So the conclusions from this part are that provision of feed for ad lib consumption leads to obesity and reproductive failure. Some hens are more susceptible than others, and it seems the ability to maintain desaturation of de novo synthesized fats is particularly important. Some expanded hierarchies, larger yolks, may be due to the availability of excess lipid and delayed release of the ovum. But we never saw the multiples like they saw in Europe. part of this is a temporal dyssynchrony between normal ovulation, and if you don't ovulate that follicle at the right time, if it doesn't go through the oviduct at the right time, you've basically lost that egg. And so if you had multiples and you had 2 yolks come out at once, for example, you would then have a double yolked egg, which you've probably all seen, but there is no room for twins inside a shell. So if you have a double yolk egg, you can't set it because you won't get a viable chick out of it. So as far as a lot of the inflammation and the apoptosis going on in the ovary, it seems that ceramide is key to that in terms of provoking that pathway, and that obesity provokes a variety of sort of non-specific inflammation that just is not a good deal. So, Jim said you guys were interested in microbiota. And I don't know, I mean, yeah?
What happens to the nervous system? You've got all this inflammation, you've got ceramide, you've got sphingomyelin. Does this do anything to any obvious changes to the nervous system or their behavior patterns?
Boy, that'd be
their
a cool
reproductive behavior?
I'm not able to ask that. So if you're interested in that, if you guys want nerves from birds like this, I'm happy to cut them out for you. It would be nice. I mean, I'm here to see if there's folks that can help me on some of this. Okay. So, obesity, it's a drag. We got to get rid of it in these breeder hens. And so, as we were sort of winding up on that story with the ceramide and all of that, Jeff Gordon and his group is coming out and saying, well, you know what? The microbiota is a new organ, and firmicutes to Bacteroides is the ratio that determines whether you're fat or not. And Canny is coming out and saying, well, you know what? If you're fat, you got these changes in your microbiota, you also get a leaky gut and you get LPS in your system, and that causes inflammation generally in a non-systematic way. It's just all over the place. And so that's why you get metabolic disease. So, you know, these were things we thought we had to look at. So here's the other thing that's sort of problematic about that, because I'll get to a point and I haven't measured LPS in the blood and you'll go, why? Well, turns out broiler breeders, one of the ways that they have selected them to grow so fast and to be so energy efficient, because they're very energy efficient, is that one of the selection programs was they injected birds with LPS. And everybody that did not show a fever response, they kept. Because if you're not spending energy on making a fever response, chances are you're going to just keep growing and not care. Okay? So, anyway, we began looking at this and seeing what was going on. And one of the questions, does everybody here know how to sequence microbiota? Is this anything I need to talk about, or can I just speed right by all this? Yes, I'm seeing yes. I'll slowly scroll through. So there's hypervariable regions, and you can sequence that and know which bug it is, because the hypervariable regions, and the reason you can do the hypervariable regions is that there are highly conserved regions that you can use as primers to bridge these hypervariable regions and then figure out who's who in the zoo. Okay. So when you do that kind of sequencing, you basically can get down to genera. If you want to go deeper than that, you've got to do more sequencing. But if you're looking at any of this Illumina, barcoding, pyrosequencing type thing, down to genera, that's it. Okay, so 2 things you have to find out. Alpha diversity, so how many different kinds of bugs are there? That's one question. And the other one is, are the bugs that are there, even if you still have, if you have 1,000 different kinds of bugs in this animal and 1,000 different kinds of bugs in that animal, are they the same or are they different? Beta diversity tells you whether they're the same or different. So the more these circles separate, the more different they are. Okay, so our question first was, well, if you put chicks in the same environment that have these really different genetics, do they just acquire a wholly different microbiota, and that's part of the reason they're so susceptible to getting fat? That was the question. And so we took these 10 broiler chicks and 10 leghorn chicks. Clearly this is just a starter one. And we put them, they were fertile eggs that were hatched in the same incubator, and then we put them into the same floor pen with new litter, and here they are standing there looking at you going, where's the chow? So we grew them for a couple of weeks, actually 4, and birds, production birds, they're very, they know a lot about their nutrition, so they give them starter, grower, finisher diets. I mean, they're very adjusted to what it is that they're going to do. And then we collected their feces individually. They get to stand in a little bucket until they poop, which never is a very long wait for a bird. And we then sequenced them, and we did their alpha and beta diversity with chyme. And then we predicted their microbial function using a little program called PyCrust, which is... So this is alpha diversity over here in the line graph, and then beta diversity over in the ball chart, the 3-dimensional chart. And what you can see is that they are not vastly different. So they seem to have the same numbers of organisms. They acquire the same numbers of organisms from the environment, whether they're a layer or a broiler. And they actually aren't too different. What seems to impact them more than their genetics is their diet. So when we go from starter to grower, you can see the clear split between blue and red. But all of those little balls, that's all broilers and layers together. So basically diet is driving their microbiota at that point. And this has been something that we've seen. Here, we are now taking them out a little bit longer. And so in this one, the alpha diversity, the only place where there was a difference was here at 5 weeks. And this is sort of interesting because at 5 weeks, they went from a grower to a finisher diet, which is much higher in energy because they're trying to get some fat on the carcass so that it's nice and juicy when you barbecue it. And so that finisher diet is far in excess of the layers' nutrient requirements. It would not be the diet that would be fed to layers normally if they were being raised for table egg production. But since this was something we were just seeing everything the same except genetics, and the broilers would really be compromised if they didn't get that diet, we gave them that diet. And so you can see that when they got all this extra goodies in terms of energy and protein, because that's what's really getting jacked up here is protein, you can see that there was a difference. And so again, it seems that diet has a bigger impact than the genetics. And over here, in these particular things, what we have is that each one of the colors is a diet, and the big balls are broilers, and the little balls are layers. And so again, you can see that the ball size is pretty much the same by color, and what's really causing differences are more the diets. So from this, there are some differences. If you get down to the general level, you can see some differences you can talk about when you try and get excited about it. But generally speaking, what we're really seeing here is that it's not the genetics of growth, it's really more the diet that they get fed. So aging intervention, even more important. Okay. PyCrust estimates based on the number of reads that it gets out of your sequencing. Gee whiz, we know this many of your bugs, and this many of your bugs have this kind of genome. So if they've annotated the genome of that particular bacteria, they say this is what it could do. You have 10,000 sequences doing carbohydrates, and so you'll get a score that way. So what I'm saying is, it is what it is, but it's not the same thing as going in there and doing transcriptomics on the microbiome. Okay, so which is what you'd want to do, really. But what you can see is that layer and broiler, there were no differentially abundant genes were detected in either of the strains at week 1, 3, and 5, but there were 22 bacterial functions that seemed to be different at week 7, where they were fed that other diet for longer. And so we have membrane transport, and this is higher in layers, replication and repair, metabolism of cofactors, and amino acid metabolism. So these are all things we would associate with the differences in the diet. I'm going way too long. Okay, so England, we're there. So it turns out cobs, arbor acres, Hubbard, these are all birds that we grow in the U.S. Ross, Avigan, is the European bird, okay? So we did our acute feeding study with these things. We swabbed the cloaca to see if a sudden release from energy restriction would cause this massive change in microbiota. These birds did something wholly different than we'd ever done before. So yes, they gained weight. They gained almost 500 grams like the other guys. Did they increase in body fat in their abdominal adipose? They did not. In fact, they had less. So really annoying. Their liver did get much, much bigger. So okay, at least that. Did their oviduct go away? Did their ovary regress? No, their ovary doubled in weight, for God's sakes. Multiple hierarchies. Different bird. Okay, so it's a strain thing. It's where are they putting the energy and how are they dealing with the excess? It's a completely different paradigm in these Ross birds than it is in the cobs. So there's some sort of genetic energy partitioning that's going on there.
I know that the generation like
Those two companies digressed probably in the 50s. They created their spots, so I don't know. And, you know, it's all pretty much by eye, how they do the selections. So it's been digressed for quite a while.
Do I understand this correctly that these are also breeder birds that you're getting like?
In terms of the ovary response, yes, completely different. But can they lay eggs? No. Well, these guys still kept laying eggs, but they would be a mess ultimately because with those multiple hierarchies, they tend to start ovulating multiple yolks, which means that you end up with a bunch of twins, which means that you end up with a bunch of non-settable eggs, which means you don't get any birds out. So you still have to restrict them, but they're doing something different when they have energy to actually play with. So that's something it's just very different. So, and you can see that their yolks get bigger, so they're probably a little bit, they're hanging on to it.
So, but they're eating a lot more and they're gaining a lot more weight, but they're not putting it into fat. Does that mean they're putting it into muscle mass?
No, they're putting it into their eggs. They're putting it into the yolks.
Double yolks.
The yolks are basically becoming almost their choice of adipose, which probably means they're more efficient at tracking liver products to the ovary. I mean, there may be differences in those lipoproteins, but and so what happened? Well, so the big thing we learned from this, the green chart on the left is increased bacterial richness, so alpha diversity increased with age in the restricted hens. So they were with us 3 weeks, and they got way more bugs. New population came in. Evidently, we have a very rich farm environment for them to live in. Now, the ad lib birds also had increases in number, but far and away the biggest impact was that we moved them physically from whatever farm they were living on to our poultry farm in the university. And so this is why when you guys move the bar shop and your mouse room back, you may have some exciting action. So yes, they did some things with the bugs and increasing the numbers. It was a little bit more as far as this, what you can see. So when you do chyme, there's a thing called unifrac. A unifrac distance tells you how far apart things are. And when you do weighted, that means that you're taking into account like which bacteria are more abundant. You're only counting the ones that hit a certain criteria. And you can see that on that case, the amount of feed these birds are eating, it didn't really matter. When we went to unweighted unifrac, where it's like, okay, we're going to just count everything that you got there, they started to separate, and they did have a significant difference. So what was really different about this, let's see, here we're looking at red and blue with amounts versus, so the weeks, the big weeks, this is the later time. So this is where all the birds came in. This was where their microbiota was, and then after time on our farm, you can see that really it's more time on the farm than it was the diet and the energy. But knowing who's who in the zoo and how much your zoo changes, even if you get more zoo members, really what you want to know is what are they up to? Because this is what's going to be determining what bacterial byproducts you get and sort of what sort of metabolites you have to cope with. And so when we looked at pie crust for the restricted birds, we saw that there were some increases in carbohydrate metabolism, cell growth. You can see these, this is kind of like a big, you know, what is it, David, and those Go annotations. It's so broad, it's like, yeah, but what changed? So we're at that level here. So you can see these are all things, okay, they got new bugs. They're doing an immune system thing, nucleotides, cool. So what happened with excess energy in addition to the environment? Okay, that's all the dark green stuff. So yes, some different things happened. These bacteria were doing something different. So folding, sorting, degradation, they're freaking out in terms of their protein synthesis. They're starting to make a lot of different glycans. We've got some sort of infectious disease, metabolic disease. Metabolism has changed. Their nervous system has changed. We have poorly characterized. Always the dream answer in any one of these sort of descriptive things. Poorly characterized. Oh, must be a grand in there somewhere. Signaling molecules and interactions. So clearly, the addition of excess energy did something very, very different to what these bugs were doing than simply, now I'm getting some new bugs to live with. The other thing that was really different about these birds compared to birds I'd worked with before, with the cob birds, if you give them food, they run over there and they eat it up. So the restricted birds, the jargon term in the industry is pickup time. Pickup time for food for these restricted breeders, if they're cobs, basically you give them the food, it's all gone inside of 20 minutes. It's gone. So they're sitting there for most of the day waiting for you to come back. So if you give them twice as much, it takes them a little bit longer, but they clean it up all at one time. They eat one big meal. Now the Ross birds, we actually had to go to split feedings because they were like, wow, look at this. Somebody's giving me like a ton of food. Wow, you know, and they were like throwing it around. I don't know if they're trying to roll in it. I don't know what exactly they were doing, but, you know, Liliana, who was running the study, came back and said, Dr. Walzem, they're throwing it. What can I do? And I said, well, I guess you're going to have to feed them twice a day. And so that's what we had to do. And they were like, they would come over and look at it. Eat some. I'm going to go over here now. Okay, maybe I'll come back. So completely different attitude. Completely different. Not this manic, rabid scarfing. But so that's a whole brain thing, people. What is different about these birds? Why? I mean, they are really restricted, and they are hungry, but why does one set run up and gorge, and the other one is like, no, I can wait. I mean, that's like my husband drives me wild. It's like, yeah, I know I've got a dozen cookies. I figured I'd do one a day for the next week. No, let's eat them all. Let's eat them all. So, anyway. So while layers and broilers acquire somewhat different microbiota, overall the differences do not account for the obesogenic propensity of broilers. Diet appears to play a large role in the divergence of microbiota and differences in predicted microbiota function. So nutrition in excess of energy and protein requirements appears to be a driver for those sorts of differences. How does this help any of me and my homegirls get over being hungry all the time? So this is the question. And so I've been very frustrated in this, in that, yeah, we can understand why it's good to restrict them, but hey, they're still restricted. They're doing skip a day. I mean, and in the industry, of course, people now are saying, well, this, and it's not the industry that's saying it, but it's a welfare issue. It's cruel to not feed birds. And so they do grants on this stuff. How can we throw the food in a way that it'll spread it out over the day, and even though it's not very much food, they'll, somehow be happy. So while I was doing some of my microbiota stuff in a mouse this time, I started working with Sean Davies. He was putting in a cell to JCI, and they were like, no, we're not going to take it until you do something with the microbiota. And so we measured some microbiota for him. And what he was doing was a very interesting thing. He genetically modified probiotic E. coli to produce N-acyl phosphatidylethanolamenes, which can be converted into N-acyl ethanolamines, which in fact are members of the endocannabinoid signaling pathway. And it turns out, depending on which fatty acid is present, if the fatty acid is arachidonate, then you have anandamide, which is, of course, homolog to THC, delta-9 THC. It makes you very, very hungry. If the fatty acid is oleate, it satiates you. And it helps you with memory integration. And it enhances your gut function. And so, learning about this, working on his mouse microbiota, I was like, wow, that's really cool. Can we feed it to these broiler hens and see if it helps them out, either in terms of reduction in spontaneous food intake, or at least in terms of not being so upset about not getting fed. That'd be nice. So we have taken this on, and I mean, honestly, this is like in the last 9 months. So we did put in a grant last year, and actually, even though we had absolutely no preliminary data, USDA, we came in 6th, they only funded to 5 out of 101. And they encouraged us to reapply when we had a little pilot data, so. But it's USDA, so who knows. Anyway, you can see all these different things, and it's not just the cannabinoid receptors that are involved, and you may remember things like riombinin that was supposed to inhibit hunger, and then people took it and they all started killing themselves, right? So this whole satiety, feeding, happiness, welfare is very much related. And so there are other receptors. There's some reductions in inflammation, enhanced satiety, and some actually, acceleration of metabolism through PPAR alpha. There's some other sort of receptors. It's all a bunch of crosstalk to each other. So this is like a new world for me too, because I'm not really a cell biologist like all of you folks out here. So we're working on it. We did go ahead and feed those old Ross hens, and we pulled some tissues and we did some primers for these things for chickens, and did some qPCR, and they do respond in chickens, so the system is there. So we're hopeful that something will come of this. And so Kristen is growing bacteria as we speak. At Texas A&M, we have birds on the ground. We are going to see if we're going to grow them out to 10 weeks and see if we can change their attitude. We did this little study for 2 weeks, and we individually housed the chickens so that we could gavage them every day and measure their individual food intake and do all that. And what we found out was it is a huge stress for chickens to live on their own. They do not like living on their own. So these are all the treatment groups. This is corticosterone. And you can see, although you told me this may be a good thing for them if it's in the morning. But so individually housed, this was their corticosterone level. This is group house. This is them standing around together. They're like, yeah, I got a flock. I'm happy. So what we mostly learned is we cannot individually house them and expect much of any kind of response because they're so stressed out from being alone. We did see, however, in this group that even though they were stressed, clearly, and during this period they're ad lib fed, so they get to choose their food. The ones that actually got the napes grew a little bit more, so they grew more in spite of being corticosterone stressed out. So is that a sign of welfare improvement? We hope so. We will tell you more. So that, I will stop at that. And thank you for your attention. And oh my gosh, took the whole time. Sorry. This is our crew. And Adam lives here now. Adam is the military guy I told somebody about. If you guys want to have military guys exercising, take leg punches out of them. They do that over there at Fort Sam. You're very kind.